him a little bit, why don't I introduce Dr. Larsen so we all get to know him a little bit more. Dr. Larsen is an evolutionary biologist with highly interdisciplinary research. Broadly, his team utilizes innovative genomic approaches, transcriptomics, metagenomics, and genome sequencing. I'm excited that I get to say those big words. And this is in advancing research in the areas of evolutionary medicine and One Health. Current projects in the Larsen lab include epigenetics of stress in dairy cattle, origin of mitochondrial dysfunction associated with neurodegenerative disease, and the discovery and surveillance of emerging zoonoses in mammals. So today we will be focused on chronic wasting disease, which I'm sure many of you are acquainted with, whether you know a little bit about it or a lot about it, or maybe you're learning about it for the first time today. So thank you again, Dr. Larsen, for joining us and sharing your research. I will turn it over to you to begin your presentation.
Thank you so much, Carolyn, and thank you everyone for joining today. Let me go ahead and share these slides here. Does everyone see that all right? Good.
Yes.
And as Melissa mentioned in her introduction, we had this amazing conversation last November. It started off focused on some of the venison testing that we do through our University of Minnesota startup company, which I'll mention towards the end of the talk. But more and more, we focused on the ecology of CWD, the environmental concerns surrounding CWD. And we just got more and more excited as the conversation went on. And so I knew that this was a group that I definitely wanted to interface with, and very happy to provide this presentation. The format, I'm going to talk a little bit at the beginning about some of the biology basics of CWD. And then I'm going to talk about some of the challenges that many of you are aware of that we're facing, mainly on the surveillance side of things. But I want to end on a positive note. There's some exciting developments that our team has come out with in the last couple of years that give new tools to fight the disease, right? And so I'm gonna highlight those and how we can use them in new ways to hopefully bring some optimism, right, in this conversation surrounding CWD. So I start almost all presentations with this question. We talk about CWD all the time. Maybe we hear occasionally that it's a prion disease, but no one ever talks about what a prion is and how it becomes infectious. And it's sort of a gap in the knowledge. And so what we've done in our team is we bring this up right away and go into the biology of what a prion is and a little bit of how it can become infectious, because it helps understand the challenges that we face with CWD out there in nature. So what is a prion? So all mammals have prion proteins. Everybody in this webinar has prion proteins right now. They're circulating in your cells. They're performing certain functions. On the right-hand side of the screen is a protein diagram. This is like a ribbon diagram that we use to understand protein shapes. And this is sort of a rough outline of what a normal, healthy prion protein looks like. So they play normal roles in cell function. They help maintain copper, zinc, and iron levels, and they're enriched. So these proteins are enriched in the nervous system. Okay, and so there's an important link there to prion diseases, right? Because the neurological function is impacted. Now, there's one way that we visualize these prions, or when we perform outreach activities, one way that's connected more than anything else that we've done over the years. And so there's a tool that we use to help folks understand, and it's a slinky. So it may seem kind of silly at first, but you have to think three-dimensionally when you're talking about prions, how they become infectious, and when you're thinking about CWD prions, because they're three-dimensional things. And this is a normal, healthy prion protein. We've used these slinkies at the Minnesota State Legislature. We've used them in Washington, D.C., and we've had great feedback on using this as an educational tool. So if you envision your normal, healthy slinky as a healthy prion protein, if I were to go to the stairs and put this down the stairs, it would function like a normal slinky would. It would walk down the stairs. Okay, so this is your model for a healthy prion protein. But when this becomes misshapen, when it changes its shape, it becomes non-functional and becomes infectious. So this is the misfolded slinky that has lost its function. It doesn't have the same three-dimensional structure as this. It's no longer functional. If I went to the stairs, it wouldn't walk down the stairs. It's lost its function. But it takes a shape that makes it infectious. And why is that? So it's the same amino acids that's in this protein, but they're the three-dimensional structure is just all messed up, if you will. Okay, so it's taken this different shape. If you have different shapes like twists in this infectious prion protein, those are different strains. And in CWD, there's about 3 or 4 strains that are across the United States. There's likely many more strains. And so there could be subtle variants in CW prions. And that can impact how they species barriers. It can impact lots of things downstream of their formation. But just know that you can have slightly different shapes that create different strains of prions. So how does this become infectious? Well, if you have the misfolded prion that comes into contact with the normal prion, let's say I'm white-tailed deer, I've consumed CBD prions, they go in my GI tract, and then they start interacting with normal prions in some of my cells, they can actually start a daisy chain reaction. They start misfolding. So all they need to do is to come into contact, and then this basically relaxes, it loses its shape, it kind of unfolds, and then it refolds in a misfolded form, and it starts growing like a crystal. Okay. So all it takes is for some number of these misfolded prions to come into, in this example, a white-tailed deer, and start interacting with other prions in cells, and then you can start this daisy chain reaction. Now, I call that the chaos engine of prion diseases. So this is happening in CWD positive animals. You have this daisy chain reaction, and ultimately it colonizes the brain. The prions are growing like crystals throughout the animal. They'll be concentrated in the brain, concentrated in lymph nodes. But it's the same thing that's happening in sheep that have scrapie. So also prion disease, also prions misfolding and colonizing the brain over time, and in mad cow disease or BSE. And in humans, it's Creutzfeldt-Jakob disease. So it's really important to understand that CWD fits in this spectrum of prion diseases. And there's a lot that we can learn about it, just knowing what we know about scrapie, knowing what we know about BSE and CJD or Creutzfeldt-Jakob disease in humans. Okay, so it's not like CWD is this crazy outlier disease. No, it's part of a family of prion diseases. So when those prions interact with each other, they can cause shape changes. So if you have a misfolded prion that interacts with the normal one, it can cause a shape change. And then it basically will daisy chain, forming crystals, if you will, and disrupting cellular function. And because they're enriched in the nervous system, you start to see neurological impacts over time. What's really frightening and interesting about this misfolded prion is that once it takes the shape, it's almost indestructible. So it can be in the environment, it can circulate in the environment for many years. These can be incorporated into plants and actually be absorbed into plant tissues. And the reason why it is almost indestructible is that it forms a dense like core. And enzymes that normally break down proteins just really don't work on it. And because it has that dense core, it can just sort of last for long amounts of time. Those prions are spread from animal to animal. Obviously, direct contact, licking. They can be the young can be exposed in fawns can be exposed in utero. So it can pass horizontally, vertically. But there's also environmental routes. And one thing that we don't talk enough about is the environmental contamination that's happening with chronic wasting disease. So if you go in some areas, as you all know, in Wisconsin, where the prevalence is pretty high, there's some counties where I think about 50% of bucks are positive in those counties. Um, you can have an increasing amount of prions in the environment over time. And so we know, and this is actually some research based out of Wisconsin by Dr. Stuart Lichtenberg, who's on our team now, that these prions are detectable in the soil at least 15 years after they were deposited. Okay, so this is one of the longest studies that's been done on looking at CB prions in the environment. Are they infectious? We still don't know that question. It could be that over time they lose their infectivity, but that's research that's going on right now. So that buildup in the environment can actually contribute to indirect exposure routes. We know that the prions can go in water. They can be transported by sediments in water. It brings up the question of, how many other animals, how many mammals are going to be exposed to prions over time, and is there a chance that it could cross over and cause problems in them? And so there's just the research community is really focused on this question right now, and there's a number of labs across the U.S. that are trying to understand, really what is the risk here. We know it's non-zero, but it's too early to say, how some of these other mammals, might react or if they could acquire a prion disease. From the human perspective, we know there's a strong barrier there. But we're concerned that over time, if you have repeated exposure events, it could actually cross the species barrier and cause a problem in humans. So just be aware of part of the difficulty in fighting CBD is that you're dealing with environmental contamination that can accumulate over time and then add to the spread. So this is a graphic of the spread of CWD across North America. The dots, and some of you probably have seen this map. This is a USGS map that they update regularly. The dots, yellow dots are, and over time, yellow dots are CWD in captive facilities, and red dots are CWD in those facilities that have been depopulated. And then the gray shading are counties that are known to have CWD. And so you can see how this is spread over time. It's likely that CWD originated in Colorado in the 1960s and then has been spreading through various means since then. So why does it always seem, when looking at that map, why does it always seem that we're years behind the disease, right? There's situations where surveillance will be done, they find the disease, and all of a sudden, as they start digging more, they figure out, well, wait a second, it's probably been circulating here for a few years. We're already looking at percentages at 3 to 5% in the local herd or something like that. That's definitely happened. And so why is that? What are the challenges with that? And there's some significant challenges with the traditional approaches to surveying CWD. Right. And most of you are aware of this. This could be, during hunting season, hunters are providing tissues to the DNR to be sent in for testing. And then that the DNR is a target number that they want to reach to better understand, what is the prevalence of disease in that particular area. So the challenge is, and the reason why we're behind the disease and surveillance is because this is a prion disease with unique features that make it really difficult to detect. And the reason why is if you are a whitetail and you've ingested prions and now you're infected, it's day 0. It can take around 2 to 3 months as those prions are sort of growing in your body, and then you'll start to shed them in your saliva, in your feces, and there's a gap there, right? So they're slowly growing. But then it takes about 90 to 100 days beyond, 120 days beyond that, so a few months before they start accumulating to the point where you can detect them in tissues like the lymph nodes that are commonly used for CWD surveillance. Okay, because you're dealing with a disease here. It's not like a virus where the virus replicates, and it's you can detect it in the blood fairly soon, or COVID, if you're swabbing your nostril, you can detect the virus with the home kit. This is more of a long process. It can take months to years. Those prions are slowly growing, and they have to get to a point where you can detect them using the technologies that folks have access to, ELISA and IHC. That progression of it can take 2 years or more before an animal that looks perfectly healthy ends in that sort of late stage phenotype where they're literally wasting away, and how the disease got its name. So that can take over 2 years, and it's variable. So this is another complicating factor to the surveillance, where you could harvest an animal that maybe has had the disease for a year, but their genetic background just caused those prions to move more slowly, right, through their body. And then because of that, maybe the concentration in the lymph nodes isn't that high, and you can get a false negative result on the ELISA test that's used for initial screening, right? Just because of the genetic background. Some animals will have a genetic background where it will go very fast, right? And you'll get a screaming hot positive, within 6 months or something. And so there's variability there just based on the individual, the populations and whatnot. There's a push now for deer genetics. Like if we can just improve deer genetics, so, we can get in front of this disease. There's some issues with that everyone has to be aware of. No deer is immune to CWD, even if you have the best quote unquote genetics. They can still contract it. And if you have a genetic background that makes you less susceptible to CWD, it's possible that you could be shedding the disease longer, right? If you are surviving that disease for an extended amount of time, you could be shedding that into the environment for a year, 2 years longer than you would if you were an animal that didn't have a susceptible genetic background. So some things to be aware of there. But the issues with CWD, the biology of the disease, the way it spreads, that adds to this diagnostic and surveillance lag time. And so it adds to the reason why if we go into an area and it maybe is only been circulating for a matter of months or less than a year or two, when you start using the traditional means to look for the disease, you could come up not detected, but it was actually circulating there. It just takes longer for the disease to get to a point where you can detect it using the traditional means. So this is part of the reason why we always feel like we're behind the disease. One of the most proactive approaches that we've seen fighting CWD is done by the Minnesota DNR. They've done a fantastic job. Early, when I joined the University of Minnesota in 2018, it was still fairly early when the disease was circulating in the southeastern part of the state. And there was a huge effort of culling the herd down in that southeastern part of the state to try to keep the disease at a low level, to not let it get to the level of it being endemic. And that's around 3 to 5% in the herd. And so using all the available resources, using the best tools that they have access to, they weren't able to keep it to a rate that it was lower than this transition phase on this graph here. And the reason why is part of the reason why there's several reasons, but one is they didn't have access to the private properties in order to go on to those sections of land and do the culling that they needed to do. And there's several reasons, but ultimately just could not get it from establishing itself as in the endemic phase. And so the sad truth that we're faced with is that the traditional tools that we have access to are just they're not quite able to help us get in front of the disease, right? We're always kind of playing catch up, and in areas throughout North America, we're just seeing these populations go into that endemic phase over time. And so the question is, how can we, with the best science that we have, help agencies? How can we provide them with new tools to be able to fight the disease in new ways? And this is where our team comes in. Back in 2019, we formed the Minnesota Center for Prion Research and Outreach, MinPro. And we had initial investment from the state. We leveraged that to start work on diagnostics. We wanted to develop new diagnostic tools, new diagnostic protocols that will give you a real-time view of where CWD is, right? If you're fighting a war, you need to know where the enemy is in real time. And so our goal was to create the tools that would allow you to see where the enemy is in real time. And we brought together this amazing team of engineers, molecular biologists, folks that are experts in hydrology, veterinary medicine. Everyone was brought together in this think tank. And we started having these amazing discoveries. A short amount of time, we had multiple invention disclosures submitted to the University of Minnesota. And in 2022, we said, okay, we have enough. We should launch a startup company called Prion. And a subset of us launched the company. And the goal with Prion is to give the public and to give agencies access to the latest diagnostic technologies that we developed. I'll talk a little bit about those. So we specialize in RT quick diagnostics, and I won't have enough time to dig deep into this, but you will, if you're up to speed on discussions around CWD diagnostics and the future of CWD diagnostics, you'll see in popular media articles discussion about RT quick. And think of this as like a PCR for prions, where we can, in a very controlled reaction, that we can replicate what happens in the animal. So remember that daisy chain event happening in the animal where these proteins or prions are interacting with each other and causing a daisy chain? We can replicate that in a test tube. And we can monitor the misfolding over time just by using fluorescent dyes. Okay, and so all RT-Quick is a test that will allow you to track protein misfolding in a test tube in real time. And on the bottom there, you see this curve. If we see a curve that goes like this, like an S curve, we can say using statistics that, okay, we believe that this is a positive sample based on this curve. If the prions, those misfolded prions are not present in the sample and they don't cause that daisy chain, it's flat. We don't see that, that curve, and so we'll say it's not detected. This test is exciting because the developer, this is back in 2010, is quoted, this is like if you took 1 tablespoon of these misfolded prions and you put it in 400 Olympic-sized swimming pools of these prions and mixed it up, this test could detect that dilution factor. So it's really sensitive. Originally developed to look at human disease, chronic Creutzfeldt-Jakob disease, and the CDC uses RT-Quick as part of their national CJD surveillance effort right now. This has been validated across several labs. USDA hasn't validated it yet, and so always when the discussion of this test comes up, it gets shuttled off into, well, the USDA hasn't validated it yet, so we're just going to have to wait for that. That's not our approach. Our approach is it's a great test. Lots of people are using it. USDA timeline is their own timeline. We don't know when they're going to validate it, but we need to use this now to fight the disease because we can use it for both biological samples and environmental samples. Okay, so we can't stand around waiting, right? This is an amazing poster that the team here at MinPro put together, and our approach all along is we need to rapidly get the latest science out into the public, out into the hands of those that are fighting this disease now, because if we keep waiting, it's inevitable. It's going to be in all over 48 states within the next 5 or 10 years. So now I'm going to transition to highlighting some of these exciting approaches that we can use right now. And in the past, I've talked about some other tests that we're designing, field deployable diagnostics, and all those are still happening. A deer side test, we have technologies that are super promising, but they're further out. The reality is this is going to take time. It could take another 2 years before we can even get some of those prototypes into people's hands. But these are methods that we can use right now, and some agencies are actually already using these methods. So state and tribal agencies can use RT-Quick to supplement their existing diagnostic tools. And then our team and our collaborators have been using these for about 4 years in both research and then in service once we started the startup company Prion. So these are the three that I'll highlight quickly. We can perform saliva-based surveillance on both wild and captive herds. We can perform environmental prion monitoring in both wild and captive herds. And we can perform prion forensics. So that is if there's skeletal remains in the landscape, if there are deer that are involved with car collisions like roadkill deer that are far beyond their tissues are degraded, we can use this test to monitor those animals as well. So these are three areas that folks are already using. The saliva-based surveillance, this is pretty exciting. So what we know based on the biology of the disease is that the prions are actually shed in saliva within about 2 to 3 months after infection. And this is controlled experiments that were done on white-tailed deer at Colorado State. And so what our team did with collaborators at Creighton University is we developed technology where you can actually swab a surface, right? Where those, let's say we know those prions are in saliva in about 3 months. If they lick a surface, right, and then you take a swab and you swab that surface and test it with RT Quick, we developed a method that facilitates that, right? So you can recover prions from stainless steel surfaces, from glass surfaces, from hard plastic, right? And so when we did that, we had this sort of light bulb moment of, wait a second, you could use this for surveillance in lots of different areas, right? It could be down in Texas, they feed deer all over the state. You can actually use this on deer feeders and see where CWD is. So on the wild side, this can be used for testing like licking branches, swabbing feeders, and also swabbing sentinels that are designed specifically for the method, which I'll highlight towards the end of the talk. And then in captive settings, it's really useful because you can swab the bunks and you can put steel plates inside the feeders and swab those as the deer are licking them. This is an example of some data that we had generated from a CWD positive ranch, and there's about 70 deer. We put 15 saliva sentinels across the pens in the ranch. And again, this was already known to be CWD positive, so it was like a positive control study. In the beginning of the work with this ranch, we could readily identify prions on the sentinels that were placed in the herd. Okay. And then over time, we could see patterns. We started live animal testing. So folks should understand that there are live animal testing options now for CWD. For what we did, we used an ear punch, saliva, and fecal sample. We live tested. We found the animals that were positive. Those were called out of their herd. And then we swabbed the sentinels again over time. And so if you use that approach, we were actually to remove the animals that were actively shedding the disease in that herd, and we got the amount of prions that were being deposited on the sentinels to be very low. Now, this doesn't remove CWD from the population. It just finds the animals that are in their, mid to late stage of shedding. It takes them out so that they're not pumping prions in the environment, right? And so at the look at the beginning, you can see those higher those red and purple colors means there's more prions there, and then towards the end we had very few signals that were actually going positive. So with that, we showed that you can actually go into a facility like that and you can identify the animals that are shedding. We can bring them out, and it's really, really important because you don't want to get to a situation like I was talking about at the beginning of the talk where you have animals just pumping prions in the environment, and then those can go downstream. They can expose other animals, and it just becomes a hot spot. So before using this technology, there's no, you have late detections, you're not live animal testing efficiently, and there's no way to really call those animals out. But using this technology, you can find them, get them out, and then keep the amount of prions that are going into the environment at a very low level. So that's really important. The second aspect, the second way that we can leverage this technology is environmental prion surveillance. And so there's been a lot of work in our team focused on soil. There's Dr. Stuart Lichtenberg is an expert on prions in soil, and he has a whole team that's just focused on developing these methods for efficiently identifying prions in soil. As a result of all the hard work that folks have been doing, there are protocols now for testing CBD in soil. We can also use this in, as I said before, licking branches, but soil at scrapes, right? And so if you go to scrape sites where multiple individuals are going and marking, and you can actually take the soil from that scrape and test it, and then you can identify, are prions being deposited in this spot right here? If yes, we need to get a team, we need to get the agency out there to do some culling right around this area, right? Can be used in natural and man-made mineral licks. And then another environmental approach beyond soil is water. And so we're just starting our work on looking at how we can use this RT-Quick technology to find prions that are being moved in waterways. So you can actually predict herds that are upstream that may be putting prions into watersheds. And so that's some pretty exciting stuff that we're working on now. But the soil testing is what's available right now that folks are using. The water testing is still in early phase development, but we see over time that it's very likely that we'll be able to test water samples routinely for prions. Okay, the third method that is pretty exciting is this prion forensics approach. And so we had a situation here in Minnesota where there were positive animals that were dumped on state lands, and we worked with the DNR to basically go out there and answer the question, like, can we confirm that these remains are actually CB positive? And we were able to do that. But as a result of that, there's these methods that we can use now, like swabbing skulls. We can actually take bone marrow and test that. We can take tissues that are completely desiccated and test those. And so what we're seeing, remains that have been in the landscape for as long as 3 or 4 years, 5 years, we've handled those and we've shown that we can test those for CWD. And so that can be used, in several ways, right? No longer, you can go now to a set of remains, and if you want to know whether or not that animal died from CWD, we can use this test to see if those prions were present, right, on those remains. So that provides another layer of surveillance. If you put all of this together and you think about how you can leverage the RT-Quick technology with the protocols that have been developed alongside some really cool modeling, because folks can model, they can take the population, the demographic information regionally, and you can model based on deer densities and whatnot, where you would expect CWD to be circulating. You could actually put sentinels on the landscape, maybe saliva-based, and test, your predictions. And if accurate, you can find the disease. You'll start to get sentinels that show that prions are being deposited, and then you can go in and strategically call. And this can be done year-round. So we think that strategic placement of CB monitoring sentinels on the landscape has great promise. We think it's a really exciting way that you can find the disease much earlier than you would traditional methods. Routinely now, there's around 4 cases now where using these approaches, we've identified the disease 1 year before the traditional methods have. So that time's really important. So, and I have a product that I'll highlight on this next slide, and I'm responsible from the University of Minnesota to declare the COI because of that. So I am a co-founder of the University of Minnesota spinoff Prion. I'm a co-inventor of those technologies, and I could have financial gain from the sale of those technologies. But this is one thing that we've developed through Prion, and we're excited about this because we're actually working with a few state agencies right now to take these sentinels, these metal housings, and you can put a mineral brick inside of them, put that on a T-post, strap it to a tree, and you can swab those. So you swab the surface of these sentinels, send the swab in, we test it, and we give the result. And so then you can monitor in real time. And so one of the first agencies that we're working with is putting these out in urban and suburban areas. They can't easily go in and cull those herds, for obvious reasons, and they want to monitor them and see where CBD is in some of those urban and suburban areas. But what we're also getting traction with is that there's organizations that want to serve as a conduit, right? So an organization that will work with their members and also be a liaison with the state agency and us to be able to do like a citizen science approach. And this gets at where that I mentioned that the Minnesota DNR was struggling because they weren't able to access certain properties and whatnot. And we think that if you had another partner come in that wasn't the agency, but was maybe something like this, where it was an organization, where it was with our company, put these sentinels in, and we can actually open up that window and see, is CWD there? If yes, we need to get in and control it. So we're talking with a few folks about how we can leverage this for real-time surveillance networks. We're running out of time. And so you've seen the map grow. We're always kind of behind the disease with the traditional technologies. We need to start using some of these new tools that have been present in the research realm now for about 15 years. We need to start using them in new ways to help fight the disease. So that we're all in this together, right? And so we have to come together to strategically fight the disease in new ways. And we really think that one way that we can do that is a proactive citizen science effort, right? That can ultimately provide the agencies with real-time surveillance data that they wouldn't normally have otherwise. So how are we ultimately waging this war over time against CWD? There's not a lot of optimism. We need to inject more optimism in that. That's what we're trying to do with these new tools. There's going to be more tools coming out over time. Public education is super important. We always have to talk the science of CWD out there. There's a lot of misinformation. We have to keep hunting. That's one of the most important tools that is available. We have to keep hunters out there in the landscape. And then what happened with our team is that we had funding come in that was strategically meant to put together interdisciplinary group. We came from different fields, different backgrounds. We had hunters part of the team to advise us on how this could actually be used in real life, right in the field. So that needs to happen more often, right? We need to have more teams formed like that, because that's how you're going to get the big discoveries that can help fight the disease in a really proactive way. And partnerships are super important. Part of the reason why I'm here talking with you all is connecting, talking about this. Are there ways that we can partner together to fight the disease? So with that, I want to try to leave some time for questions. Thank you so much for reaching out to me initially, Melissa. It was a fantastic conversation. And thank you all for joining today. Happy to answer questions.
Wow, I learned so much. Thank you deeply, Dr. Larsen, and thank you to all of you for being with us. So as we pivot into the Q&A, I have a few ready to go in the chat, but please feel free to add a few more. And then stick around for our closing right around 5:25-ish. So our first question comes from Michael Miller. He asks, do you have stable funding research that will allow you to continue your learning?
That's a great question. So do we have stable funding? One thing that has sort of insulated us from what's happening at the federal level, because there's some serious concern at the federal level for federal grants that will be available, in the coming months and years. But because we've been largely state funded, we've been protected from that for the most part. We just submitted a renewal for our research program, and we have great relationships with legislators on both sides of the aisle. And so we fully anticipate that we'll have another 4 to 5 years of funding awarded to us next year. Great question. We're riding this wave. Could we use additional funding for certain areas? Absolutely, always. right? We want to expand our plant research, right? So we need funding specifically to go in to help us look at prion uptake in plants and how can we prevent that from happening, for example. But thank you for your question.
Thank you. That's encouraging to hear that you have that funding
Mhm.
secure. So Michael Samuel asks, could you elaborate on false positives and estimating prion concentrations instead of presence absence?
Yeah, absolutely. So one thing about RT Quick is that folks, for whatever reason, I don't know where this came from, but when you're working with tissues, let's say you're working with lymph nodes, just like you're working with ELISA and IHC, the traditional tissues, the false positive rate is the same or less than what the false positive rate is on ELISA and IHC. So we're really not seeing an increase in false positives with RT Quick when you're using, brain and lymph node. Um, the false positive rate can go up a little bit when you're looking at environmental samples, but the beauty of the method is that you can do repeated sampling, right? So it's not like you're one and done. You can go back and you can resample and you can see if you get the same result, right? All of the testing that we perform, it's the same with ELISA IHC, same with RT Quick, is that you're using controls, positive and negative controls. So false positive rate is in line with the currently available tests. The false negative rate is what we're worried about with the traditional tests, right? And so we see an increase in false negatives with ELISA and IHC, but RT Quick is sensitive enough to catch that disease a little bit earlier, right? And so the false negative rate is actually a little bit better than traditional tests. But it's a really important question. There are strengths and weaknesses to every diagnostic assay, but if you use them together, right, it's super powerful. And those rates do change based on the tissue you're testing, and whether or not you're using environmental samples.
Thank you. Could you comment, this comes from Dale Baker, can you comment on susceptibility of other species, like humans,
Yep.
to deer prion disease?
Yep, great question. And so all the available data that we have would indicate that there's a strong barrier to CBD from humans. So it's not that it's not like mad cow disease or BSE, where something like 300 people, died by consuming those cow prions. This is different. There's no evidence that we see of like clusters or outbreaks or people getting Creutzfeldt-Jakob disease from consuming positive venison. The CDC is monitoring this. Um, we know on other species like cattle that some cattle have a particular genetic background where they will contract a prion disease when exposed to CWD. Those are controlled experiments that the USDA is running. The same with sheep. CWD can cross over into sheep and cause disease. Some percentage of pigs in controlled experiments in the USDA, something like I think around 20% of pigs in their challenge experiments did contract a prion disease. So we know that there's potential for other species to contract a prion disease from CBD. What we don't know, and this is where the scientists, like where we get concerned, is that the shape of prions in Wisconsin may look like this, but the shape of prions in Colorado may look like this, and the shape down in Texas may look like that. When you have the shape variants, does one of those fit really nicely with the human prion or fit really nicely with the cow prion? Maybe. And so that's where the concern is that we don't want to test that. We need to stop prions from getting into the food chain, animals and humans.
Thank you. Isn't the surveillance system likely to spread CWD similar to baiting or feeding? Could you
Yeah, we
speak to
that
that?
comes up all the time, and there's a trade-off there. Like so it takes about 10 milliliters of saliva to transmit the disease, right? And it really comes down to what the agencies that are using it are comfortable with. So we know, like down in Texas, that feeding is so ingrained in the Texas culture they're never going to stop. It's just not going to stop. And so, using those sentinels down there, um, excited, they wanted to use them, right? Same in, there's other regions in the South where that culture is there. When the targeted culling approaches that the DNR was using here in Minnesota, when they were performing those in January, February, March, they use baiting as part of that, right? To bring the animals in. And so, we understand what that concern. If you swab and it comes back positive, then right? You can take down the sentinel, but it could give the agency a year or 2 years of a heads up, right? So what are you comfortable with, right? But good question.
So you've talked a bit about the potential of citizen scientists and community involvement. What else can citizen scientists do? Campers, hikers, etc. Can we help with collection from the environment?
Yeah, I mean, there's definitely opportunities there. The main thing is, what if we're going to have really have an impact, there's got to be a way where it's structured, right? There's a structured approach where the data are collected and maintained in a certain way to maximize that. If we're collecting these data and we work with the agency to give them those data, it's got to be solid so that they know how they can use that in real time. So how folks can help with that? There's going to be opportunities, I would say, where they could be part of a formal program where there is structured data collection, right? And they can participate in that. We're just early on in that process, right? Right now, what folks can do is if you see it's rare to see an animal that's looking like the end stage phenotype, but it does happen. There's a case here in a small town in northern Minnesota where there was a end stage CWD animal that actually died in their front lawn. So you look for cases, look for animals and that may be acting strange and report that, right? Keep hunting is another way that the public can help. And make sure that you're aware of the CB laws and regulation, right? Education is so important, right? And so that's part of what we're doing is outreach at events all over the state and beyond.
Thank you. We have just another minute or two for questions. So, uh, in a few words, what does selective culling look like once the prion is found in saliva?
Oh, so so just I'll repeat so I can understand. So if we find, right, that one of the sentinels goes positive, right, what is the next step that the agency would take? So, in our experience, it would be sharpshooters, right? If the area was amenable to that, right? It's going to be different depending on where you're at. So if we're here in St. Paul and a signal goes up, the response is going to be different by the agency. But in a sort of traditional natural setting, if a sentinel goes positive, and maybe you get two swabbing events that shows, yes, this is coming up positive now, several swabbing events in a row, then what would happen is that the agency would come in with sharpshooters and they would try to selectively remove those animals from the landscape. And then we could test those and tell you whether or not you found the animal in that area with CWD. After the culling event, you could have the sentinel still remaining and see if you're still getting prions or not, and that could help you understand if you got all of them.
Thanks for illustrating that a little bit more. So as our last question, this comes from Jerome Donahoe. What is the basic mechanism that causes a normal prion to misfold to that misfolded prion
Yeah,
protein?
that's a great question. I would say scientists are still trying to fully understand that. There's a few different hypotheses, and it could be that like in the cell, if this is happening, right, they start coming together, and some folks think that there's other cofactors, other molecules that will come in and then start unwinding this, right? In the presence of this molecule. Uh, and what happens is when it unwinds, it basically clicks like a magnet on top of each other, and it just happens like boom, 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 and it keeps growing. But the actual molecular mechanisms that drive that, still not well understood. But this is where I get excited about when thinking about, okay, let's develop a vaccine, or let's get a therapeutic out there to help prevent this. You can think of a blocking molecule, right? So if you were to engineer and develop a molecule that would block that from happening and get that into the animal, that might be a way so that you could actually prevent the spread. And there are folks that are working on things like that globally. Mhm.
There's just so much exciting stuff to learn here. I know we're all muted in our different places around Wisconsin and elsewhere. Could we give a quiet round of applause to Dr. Larsen for this presentation?
Thank you, everybody. Thank you so much. And always happy to answer additional questions. Please, Melissa, Carolyn, feel free to share my email address to folks. And I think through presentations like this and our outreach events, we've reached over about 150,000 people now through like direct one-on-one events, state fairs, things like that. So thank you, everybody, so much for attending today.
That's amazing, just the amount of outreach. It's such an important topic. I would like to invite everyone to please take this follow-up survey. You can scan the QR code, and in a moment, I will put this in the chat. We will also be following up with you via email. The presentation has been recorded, and we'll be providing that on the Wisconsin Screenfire website. I really encourage all of you who aren't already members, thank you to those of you who are, to join us for more presentations like this, for other casual gatherings. We need one another right now, and if this has been meaningful to you, please join us in this work. So thank you again, everyone, for being here, to Melissa for making this happen through that initial conversation, to Cody and all of you from Wisconsin Wildlife Federation. Truly appreciate being together in this webinar. And to Dr. Larsen, not only for sharing all this information, but for that invitation to stay connected.
Yeah, absolutely. Thanks, everybody. It was a pleasure. Thank you.
I'm going to